here we are trying to show once again the same thing, but this time it is yellow with green. So this is 2 alleles, same alleles yellow, and here we are having green. You know, already we have studied the meaning of the terms alleles. So 2 alleles are same, here these 2 alleles are same. So this is homozygous, this is also homozygous. Now the resultant offsprings that we get because of the crossbreed between these 2 plants, we call these plants over here, the resultant plants, as offsprings. So these offsprings, when you try to look at, you have that these are having 2 different alleles. One is coming from here and the other one is coming from here. So this is heterozygous condition. We in the previous session learnt what are heterozygous alleles and homozygous alleles. So same thing what we have studied over here, cross between yellow and green. Let us try to understand with the help of pictures. So here is a pea plant with a yellow color seed, and this is another pea plant with green color seeds. Crossing between them, breeding between them is shown using this into mark, and the resultant is shown using this arrow, and you get the new plants over here which are offsprings, or we call it as also progeny. Now when these plants grow, crossbreed, these are hybrids, when these plants grow, what color will you get for the seeds? Do you get yellow? Any idea? Or do you get green? What color seed will be produced by these offsprings? Because this is heterozygous plant, it is having half of this and half of this. So which color seed will be developed? Dear student, here we can find the plant producing yellow color seeds. Yellow color seeds are produced. When you are talking about the cross between 2 different plants with yellow seed and green seed, what color seed is actually achieved in F1 generation in offsprings? It is yellow color seed. Again, let's talk about the important terms related to this particular breeding. So here pea plant, what we have taken, this is pure plant, so we call it as pure breed over here. When you say pure breed, we are talking about homozygous alleles, both the alleles are same. Next one is the green plant. Here this plant is green plant, so that is also again pure breed, because this is again homozygous, alleles are same. But what about the generation that we are talking about, first generation, the offsprings that we achieved over here, these offsprings we call them as first filial progeny or first filial generation, the plants that we attain because of crossbreed, also termed it as F1 generation, F1 plants. So what is that we achieved in our crossbreed over here? Same once again, here I have shown the crossbreed using plants. We are showing the same thing over here using symbols, seeds with the color. So this is yellow color plant with the yellow color seed, this is green color plant with green color seed. Cross between them. In the first generation, the offsprings, what is the color of the seed that we achieved or Mendel achieved for that matter? They were all yellow color seeds, the plant with yellow color seeds. This is first filial generation. Dear students, do we mean to say that the green color nature is not there in these plants? In this, it is there, but green color is hidden, it is not shown, it is concealed. So what is happening here? Only yellow color is expressing itself here. So yellow color is dominant, it is not letting green color to show. That means whenever you go with a crossbreed, that means if you are talking about 2 different individuals which are differing in one character, whatever individuals you get over here, you find that only one of them will show their character, the other one is not expressed. This is the first Mendel's law, which is called as law of dominance. We will see this once again. So let us talk here the same thing, trying to show where the alleles can be present and how we can show once again the formation of the gametes using square boxes. This is called as Punnett square box. This is the Punnett square box. Now here these 2 are the alleles. These 2 are the alleles representing yellow color plant, pure plant, and these 2 are the alleles of green color plant, that means green seed. Green color means I am talking about the green seed. So these 2 are the alleles. Now combination of this with this, that is capital Y and small y, yellow with seed. What color plant did you get over here? It is yellow color seed. Again this Y over here with this, then what did you get over here? Again it is yellow color seed. So like that, over here also yellow, over here also yellow. So here you get all F1 plants, the offsprings because of the crossbreed, all of them are yellow in color. Though green has participated, green did not show, it did not express itself. This is called, dear students, law of dominance. One color is dominant, not letting the other to show. So here is Mendel's law of dominance. Mendel's law of dominance states that in a heterozygote, why I am saying heterozygote? Now let's come back to this. What about these two alleles? This is a plant which is producing yellow color seed. This is another plant, pea plant, which is producing seeds of yellow color. Again plant with yellow color seed. This is fourth plant with yellow color seeds. All are the plants with yellow color seeds, right? But if you look at these two alleles, what about these two alleles? Are they same here? One is capital Y, that is of yellow color, and the other one is small, that is over here. This is nothing but hybrid. So hybrid plants have different alleles, which we call as heterozygote. So dear students, Mendel's law states that in a heterozygote, one trait will conceal, will hide the presence of another character, another trait, another character, right? Both we are talking, yellow is also talking about the color, green is also talking about the color, but only one is expressing itself. So both of them are not contributing here to the color of the seed. That is the reason we call this as law of dominance. Now if you are talking about this FN plants, these are all FN plants, all are the plants with yellow seeds. Suppose if you are talking about selfing of these, right, then that is called second filial generation. If FN generation plants are allowed to go with self pollination, which we call it as selfing, then we get F2 generation. So what happens, what will be the plants, the color of the seeds in future in the F2 generation? Let us see over here. So we'll come again from the beginning. We are having again over here parents, these are the parents, right? Pure plant, that is plant with yellow seed, both the alleles are same. Again here this is green color plant, this does not show in the first generation, that means this is recessive character, it is not allowed to show. So this is having green color, so this is recessive character, which we represent it as YY. Why are we saying this is represented as a small letter y? It is because this is here yellow is dominant, so dominant color we use the letter of this dominant character as to show that particular gene or that particular allele. So here yellow is dominant, so we take letter Y as allele, and that is dominant. So this is pure breed yellow plant, this is pure breed green plant, in the sense the color of the seed. Now when we crossbreed them, right, monohybrid cross we are talking about, crossbreed them, so these are the offsprings we get, all of them produce yellow seeds. So this is first filial generation. Now when these plants are allowed to go with selfing, self pollination takes place, here you can get even green color seeds, that means plants which will produce green color seeds. This is the result of F2. This is the result in F2 generation. F2 generation, you see yellow as well as you see even green color seeded plants. So the same thing, dear students, what we are showing over here, right? Plants, then progeny, this is F1, then again selfing F2, same thing we are representing here with the help of this diagram. Okay, so yellow seeds, capital Y, these are the two alleles. These two alleles, of the two alleles only one will enter always whenever there is gamete formation, these two will not enter. So from capital Y only one allele, one capital Y is entering gamete. So similarly over here these are two alleles small y, out of this only one enters this gamete, one allele enters the gamete. So cross breed takes place, that is self pollination occurs and you get F1 generation. F1 generation all are yellow seeded plants, right? On selfing, selfing means self pollination takes place. So how do you represent that selfing over here? So, this is capital Y, small y, again into is breeding, right. So, capital Y, small y into again capital Y, small y. So, one of these parents is represented here, this capital Y is this small y is this. We are talking about selfing. So, if this represents the female part, then male plant is represented over here as capital Y and small y. Now, dear students, what about this? I am talking about F2 generation. What about the plants over here? So, here there are 4 boxes. So, you get 4 plants over here. Now, here let us look at the color of the seeds. So, here this is yellow color seed because this is capital Y, 2 alleles, capital Y, this is yellow seeds. Now, here this is small y, this is capital Y. So, this is capital Y, small y, means again you are getting yellow seeds. Why? Because capital Y is dominant. So, though green is present, it is not expressing, it is recessive. Next, third box is showing this gamete with this gamete, right. So, here we have capital Y, small y, again yellow seeds. What about this, dear students, in the fourth box? This is small y and this is small y. The hidden character, see, green is not exhibited here, but it is concealed, right. Now, it is showing off itself in F2 generation. So, small y are these green seeds. This is homozygous, both alleles are same. That means, in F2 generation, we got 3 plants with yellow seeds and we got 1 plant with green seed. Now, dear students, I have shown the same thing over here again with the help of another diagram. We are trying to take once again over here parents, pure plant, over here pure breed. So, here again yellow plant, yellow in color. These are the 2 alleles, both the alleles are same. So, homozygous alleles, these 2 are same homozygous alleles. So, pure green, right. Now, this is hybrid. Because of the breed between them, you get this is a hybrid. Here the alleles are different. So, this is heterozygous condition. Now, here the plants that are produced, they are all yellow color plants. Now, if you talk about on selfing, right, capital Y, small y into capital Y, small y. So, one side is representing parent, father, we can say, the other one is the female part. So, when you go with selfing, then you get here 4 combinations as I showed over here, same box over here. With the help of color, it is easy for us to understand. Hence, you can see over here that there are 3 yellow color plants, that is plants with the seed yellow color, and one is green. See here, yellow seed, capital Y. In the same way, yellow, capital Y. This is again capital Y, small y, wherever capital Y is there, that is dominant, and this is small y, this is green color. So, we got 3 yellow color and 1 green. So, on selfing of this, we got 3:1. Now, dear students, whatever combination we got over here, the same thing we can try to express it using 2 more terms. What is it? One is called as phenotype and the other one is called as genotype. What is phenotype? Phenotype means appearance wise. See, you are saying that we got here how many plants which are giving you yellow color seeds? 1, 2 and 3. So, how many over here? 3 yellow seeds we got. Plants with yellow seeds, they are 3 in number. There are totally 4 plants we achieved, out of which 3 are the plants which are showing yellow color seeds appearance wise, which is visible to us. What about green seed? How many did we get the plants over here with green seeds? Only 1 we got, right? 1 is visible to us. So, phenotypic characters or traits means which is visible to us, the qualities which we are able to see, that is called as phenotypic. So, yellow seeds versus green seeds, if you look at the ratio, you get it is 3:1. This is phenotypic ratio. The crossbreed, mono-crossbreed ratio, right, of yellow seed with green seed, we get the ratio as 3:1. 3 are yellow seeded plants and 1 is green seeded plants. But let us look at the genotypic ratio, right. Are the alleles the same? Genotypic ratio, we are talking about the presence of the alleles and whether they are same or not. So, look at this over here, this is capital Y, this is capital Y. So, how many capital Y we are having over here? Capital Y, we are having only one capital Y. What about this? This is capital Y, small letter, small y. This is capital Y, this is small y. So, how many are there? Capital Y and small, that is heterozygote condition. These are 2 in number. Heterozygote condition is 2. This is the hybrid that we are talking about, 2 in number. What about this green plant which is y, small y? How many such combinations we got? Only 1. So, genotypically when you see, look at the types of the alleles, pure alleles, homozygous alleles, you find homozygous dominant 1, capital Y, homozygous recessive which is pure yellow, small y 1, and you get 2 heterozygote, capital Y, small y, you get 2 such plants. So, this is the genotypic ratio which is 1:2:1. Same 4 plants we got here, right? 1:2:1. 1 pure yellow, 1 pure green, 2 yellows, but you have even green color in them hidden. So, this ratio is called 1:2:1. 1:1. So, 25% of them are pure plants, 25% pure green, pure yellow, pure green, and 50% is hybrid plant. So, this is the difference between phenotypic ratio and genotypic ratio. Dear students, we have noticed one thing, that during this F1 formation, that is offsprings are produced because of the crossbreed, these 2 alleles, they did not blend, they did not fuse, they did not mix up to produce some other character. These 2, though yellow showed, we find that these 2 alleles did not mix up. That means they maintained their distinct character. So, next time when alleles were formed here in F2 generation, each gamete, the alleles were separate. So, that is called as the second law of Mendel's inheritance, which is law of segregation. So, what is law of segregation? The 2 alleles of a gene when present together in a heterozygote state. Heterozygote state means we are talking about F1 plants. So, in F1 plants, when the 2 alleles are present, they do not fuse, they do not mix up, they do not blend, but they remain distinct. And next time when the gametes are formed, these 2 alleles get separated out and individually alleles enter into gametes. This is law of segregation. Now, let us try to understand law of segregation in terms of pure and as well as recessive, homozygous dominant, homozygous recessive, and here in heterozygous condition what happens. So, if you are talking about homozygous dominant Y and Y, so gamete will get, one gamete will get either this capital Y or this capital Y. In the same way, homozygous recessive, each gamete that is formed will get recessive allele, this one or this one. See, in heterozygous nature, here F1 plants that we referred to previously, here the gamete can get either capital Y or it is recessive allele, small y. So, here in heterozygous condition, these 2 did not mix up to produce some other trait, they maintained their distinct nature and go separately in gametes. This is called law of segregation. Now, if we go with dihybrid crossing, what does it mean? That means we are talking about here crossing of 2 individual plants, 2 plants which are similar but differing in 2 characters. So, such a type of crossing or breeding that takes place over there between these plants is called dihybrid cross. So, Mendel chose over here for dihybrid crossing to study. He has chosen color of the seed of pea plants along with the shape. So, he found that yellow color seed, which we already saw a dominant character, that one, and the shape of the seed was round, that he has chosen. So, these 2 are dominant characters. So, green color seed is recessive, we know that, and shape of the seed over here is wrinkled. That means outer shape is not round, it is wrinkled, right. So, this is pure plant, this is pure plant. In F1 generation we find all round character is dominant. So, we get over here in F1 generation all yellow and round seeds, but even wrinkled character is seen, green is also present but hidden. This is F1 generation. F1 generation, when we go with selfing, self pollination, then we get different combinations. If you look at the combinations, we get round and yellow, we write dominant nature as capital R and capital Y for yellow color, round and yellow, round and green, wrinkled and yellow and wrinkled and green. These are the 4 combinations we get. So, the ratio is phenotypic ratio, appearance wise it is 9:3:3:1. That means for dihybrid ratio, we are not studying genotypic ratio, we will study that in the higher classes, but phenotypic ratio of dihybrid plants is 9:3:3:1. So, wrinkled and green, wrinkled, pure wrinkle and pure green, the parent plant what we started with, we get only one plant in F2 generation. Same thing, another easy method to remember this one is, suppose I am going, if I go with 1, 2, 3, 4 like this plants, if I go with laming. So, if I try to connect the, if let us say that all these boxes have a center one dot. So, if I connect all these dots of yellow like this, I can get one triangle like this. Count all this 1, 2, 3, 4, 5, 6, 7, 8, 9. So, this is one triangle with 9 yellow color, yellow and round. Then another you can connect one dot from here from green to this and get another triangle over here. So, these 3 showing 3 green and another one over here in the last here, this is a small 4 boxes, we can show one dot, one dot, one dot and try to connect, we get one triangle. So, this is again yellow color, right. So, what did we get over here is that we get the same thing, round and yellow which is 9 and round and green, these 3 are round and green, 3 in number, then here wrinkled and yellow 3 and then pure green with wrinkled 1. So, like this we can remember the phenotypic ratio of a dihybrid cross, which is 9:3:3:1. Dear students, we have studied that chromosomes are made up of genes, genes are made up of DNA. So, research of GNA of a gene being made up of DNA, these are the 2 great scientists over here, you can see they are James Watson and Francis Crick. So, these people gave detailed study about DNA. DNA is a material that actually makes up genes. So, let us come back to this. We started with this particular chromosome in our previous session, chromosome making up gene, gene making up DNA. So, what is DNA? DNA is nothing but double helical structure. So, just imagine that there is one snake in one direction from anterior to posterior and another snake in inverted position, both the snakes twined up together, right. So, whatever is the shape you get, the coiling, that particular shape can be called as double helical structure. Or take any 2 ropes, one rope from top to bottom and another one around it in reverse direction. Similarly, DNA also has got the double helical structure. So, what is this DNA again made up of? DNA has got one sugar molecule over here. The full form of DNA is deoxyribonucleic acid. So, ribose is this sugar, pentose sugar, which is attached to one phosphate molecule, and to this sugar molecule is attached actually one chemical which is called as actually nucleic acids. There are 4 nucleic acids which are present. These 4 nucleic acids, this is one chain, another chain runs in opposite direction to this. So, now here what are these 4 nucleic acids? They are given different names, 4 different names. One is adenine, second one guanine, third one cytosine and fourth one is thymine. Now, the pairing A always goes with T. Adenine always forms bonding with T, thymine, that is double hydrogen bonds can be seen. Cytosine always bonds with guanine, again using triple bonds over there, glycosidic bonds and hydrogen bonds are present, right. So, A always pairs with T and G always pairs with C, always the pairing is fixed like this. So this combination gives rise to part of DNA which is nothing but making up one gene, right. So it is not that always A is present over here, it's T is present over here, it's not like that. A can be present here, then obviously its complementary is T is over here. Similarly, if G is present, C is present here. If A is present, then its complementary T is present here. So it goes on. This is nothing but giving you the code for making proteins that is needed to run our body. Now dear students, let us try to understand how we can determine sex in human beings using these chromosomes. Already we know that we have learnt we have 23 pair of chromosomes, 22 pair of chromosomes are autosomes, and the next last pair is sex chromosomes. So if you talk about man, in man we have 44 chromosomes, right. 45th chromosome is X chromosome and 46th chromosome is Y chromosome. So X and Y are sex chromosomes. Similarly, if we talk about a female, she has 44 chromosomes which are autosomes, 45th and 46th both the chromosomes are same, that is X chromosome. So in case of females, ova has only one set of chromosomes and the sex chromosome that is getting into this is X chromosome. But the sex cells produced by man, which are sperms are of two types. Half of the sperms carry X chromosome because there are two sex chromosomes. Half of the sperms carry Y chromosome. Now, if this ovum with X chromosome, it fuses with this sperm which is having X chromosome, then the child is a baby girl. If this ovum with X chromosome, it fuses with the sperm which is also having Y chromosome, then the baby would be a boy. X combination with Y gives boy, X combination with X gives rise to girl. So using this chromosomes we can determine the sex of the child. Now dear students, we have a very small activity to understand how actually the alleles, though they stored together, when it is a time for them to get into gametes, how they get separated and get into the gametes. Let's see that. Dear students, we have a very small activity which will help us to understand how actually alleles are present in a gene together, and when it is the question of gamete formation, how individually they get separated out and go, which is nothing but again law of segregation. So what we need to do is take two containers. That means one of the containers represent female and the other one represents a male. So over here you can see that this is representing male and this is representing female. So here I am taking over here, these are the seeds, let's talk about. So how many seeds I am taking over here? Yellow color seeds 1, yellow color seeds 2, 3, 4 yellow color alleles, and then green over here 1, 2, 3, 4. So this is having now 4 yellow colored and 4 green color, let's say alleles together. And in another cup same thing over here, yellow 1, 2, 3, 4 and dark green color 1, 2, 3, 4. So now let's talk about crossing of this, right, selfing of this, because we are having the same alleles here, right, at a given time. Now if I pick up one from here, one from here, what type of combinations will we get? So we are having over here capital Y, small y, that is yellow and green, here also capital Y and small y, yellow and green. Now if you take, what kind of combinations we get? How many times we will get yellow with yellow, how many times we will get yellow with green, and how many times we will get green with green? Let us see that particular number and the chances over here. So first I am removing from here, taking out one. Let us say this is green and this is yellow. So first time we have seen yellow and green coming out, dear students. Let us see now second time. Second time again we got yellow and seed. So let us make a note of this. Again second time yellow and seed. And now let us see the third time, right? Third time again yellow and green, right? So again yellow and green third time, right? Now fourth yellow over here, yellow and yellow combination we got for the first time, right? Then again let's see what is this. This is again yellow and green. What is this? Green, green and yellow again. This is what? Again yellow and green. This is green and green once. So dear students, I have taken here 4 green and 4 yellow, 8. 4 green and 4 yellow, that means 8 totally, right? So let's count this. Yellow plus yellow combination we got once, green plus green we got once, right? So 1, 2, 3, 4, 5, 6. 6 combinations we got wherein yellow and green were together. Like this, you find that the alleles are combined in heterozygous condition, and during gamete formation they have a choice to get separated out and move. I hope you understood this activity, dear students. You can do this with various other things. So finally, whatever we have learnt, dear students, so far about the transmission of characters, variations, heterozygous conditions, all this is nothing but study of genetics. Study of genes, study of heredity, genetic variations. This is called genetics. What we have learnt so far in our previous session and this session, if you try to sum up, it is all about the difference between heredity and inheritance, about heredity, genetics, Gregor-Mendel's contribution, chromosomes, what they are made up of, genes, homologous, heterologous chromosomes, alleles, homozygous and heterozygous alleles, what is monohybrid and dihybrid cross, what is phenotype, how is it different from genotype, and we also performed an activity which with the help of which you can understand how alleles get separated out. Better understanding and practice, you try to download worksheets from SCERT website, try to solve them, have discussion with your friends, and for any doubts ask your teachers. Thank you very much.
सबको सादर नमस्कार। प्यारे बच्चों, आज के इस डिजिटल क्लास में आप सबका फिर से हार्दिक स्वागत है। आज जो हम पाठ पढ़ने वाले हैं, यह ऐसा पाठ है जिसका संबंध गीत और संगीत से है, प्रांत और समाज से है, परंपरा, संस्कृति